this lesson on artificial intelligence in sciences. Bacterial infections have a large impact on public health. In the past, bacterial infectious diseases were uncontained, causing epidemic outbursts throughout the world. The discovery of bacteria in the mid-19th century was a game changer for improving public health. Since then, detecting and characterizing bacteria causing infections has become paramount. Automating this process using computers can be a game changer. This unit provides an overview of the application of AI for bacterial image analysis. Let's get started. The exploration of microorganisms and microbial communities involves a collaborative approach across molecular biology, biochemistry, and biophysics. Microbial cultures are observed using specialized imaging systems involving at least a light source, a set of lens, and a camera. Following image acquisition, we want to process its content for further analysis. The goal is to automatically predict microbial community behavior, enhancing our comprehension of microorganisms' behavior and interactions. Some of the challenges associated with this process are segmentation, object detection, denoising, and artificial labeling. Cell segmentation is vital for quantitative analysis of microbial colonies, as it provides detailed insights into cellular structures and behaviors. This can be very useful for a variety of applications, such as analyzing the morphological characteristics of individual cells or tracking the development of cell lineages over time. For cell segmentation in microbiology, we can use deep learning algorithms such as UNet, CARE, Pix2Pix, StarDist, and SplineDist to locate individual cells and outline their shapes. In the image, we can see the segmentation of Escherichia coli bright field images using the models CARE and Pix2Pix. Results of segmentation can also be used for dynamically tracking individual cells in a video feed to observe their behavior, like in this still frame of Escherichia coli cells, where they are being segmented using StarDist and tracked using TrackMate. Object detection, distinct from image segmentation, assigns bounding boxes and class labels to detected objects. Why is this useful? For example, if you want to estimate the density of some kind of cells in a given sample, irrespective of their shape, you first need to find them, right? And if there are different kinds of cells present, you need to distinguish them, of course. In microbiology, object detection classifies cells by growth stage and performs synabiotic phenotyping, aiding drug discovery. In this example, a YOLO model was utilized to detect and classify different growth stages of live Escherichia coli cells. In the context of imaging, phototoxicity means that the light used to observe or study cells can sometimes cause damage to the cells being observed. This is particularly relevant to techniques like fluorescence microscopy, where intense light is used to visualize cells, and if not managed properly, it can affect the health and behavior of the cells, potentially leading to inaccurate or misleading observations. That is why lower light doses are commonly used for this purpose. There is a caveat, however. Lower light intensity produces noisier images. So how do we compensate? We can do this by using image denoising algorithms like Pure Denoise, Noise to Void, and CARE. We can observe images of live Escherichia coli cells with low and high signal-to-noise or SNR ratios and the results of algorithmic denoising. Artificial labeling is used to improve the quality of images in scientific research. It involves using an algorithm to predict details in an image that are not directly visible. For example, in live cell imaging, it can create colorful images from black and white ones, helping scientists see cells more clearly. This technique doesn't require manual effort to label images, making the research process faster and more efficient. Artificial labeling is especially useful for studying tiny living organisms, like bacteria, under a microscope. In the image, we can see the results of artificial labeling of Escherichia coli membranes using some popular techniques. In conclusion, the integration of machine learning techniques in microbial studies offers valuable solutions to various challenges in image analysis. From segmentation and object detection to denoising and artificial labeling, these approaches enhance our ability to extract meaningful information from complex datasets. The application of deep learning algorithms, exemplified by models like UNet, CARE, Pix2Pix, StarDist, and YOLO, contributes to the advancement of understanding microbial behaviors, interactions, and patterns that may otherwise be challenging to discern through traditional methods.